We will be beginning the cell respiration lab tomorrow. We'll start by getting used to the setup and we'll run it on Friday. This is just a quick pictorial representation of what we're going to be doing. You'll have 3 vials that you're going to put together. One of them you're going to add germinating peas that have had water added to them and they're starting to undergo the process of growing. The other vial, you're going to add non-germinating or dry peas and some glass beads so that you equal the same volume as your germinating peas, which are bigger. So the glass beads are to control for volume. And in your third vial, you'll have glass beads alone because they are going to be our control. The little air spaces in between them will represent changes not due to metabolism, not due to oxygen consumption. And so we want to be able to subtract that out. Those are just changes in gas pressure because of temperature changes, or changes in gas volume because of temperature and pressure changes. So hopefully just that little intro will help you when you're doing the pre-lab understand what this table is, because it is a bit confusing. You are going to be yourself either doing a room temperature bath, which is 25 degrees Celsius, or putting your vials into an ice bath at 10 degrees Celsius. So you'll be doing one of these and you'll be sharing your data with other class members. For the purpose of the pre-lab, you're filling out the data table as if you're doing everything. So at 25 degrees Celsius, we have readings for the vial that has beads alone. We have readings for the vial with germinating peas. And we have readings for the vial and we have readings for the vial with dry peas and beets. And we have time intervals, so we're reading at time 0, at 5 minutes, and at 10 minutes. What the 0 to 5 or 0 to 10 means is that we're going to take a cumulative reading, though, so that at the end of 20 minutes, we'll see how much change there's been, how much oxygen has been consumed. You'll be reading the pipette. And so if it says 0.93 at time 0, and it moves to 0.91, and it's going backwards, which is just the way that we're going to be reading it. But if it moves to 0.91 at 5 minutes, then there's been a difference of 0.02 mils. And that is literally just taking this number, subtracting it from this number. Now, we know there's only glass beads in there, so that difference in volume is not due to metabolism. So we have to subtract that out from our other vials. So for germinating peas, if this is where the starting measurement is, and this is where it is at 5 minutes, when we subtract that out, that's a change of 0.07. However, we know that 0.02 of that is not due to metabolism. It's just due to pressure and temperature changes. And so we have to subtract that out and say that really the amount of change due to metabolism is only 0.05. Okay, so this number is the corrected difference. We get that from taking this number and subtracting the beads amount. Okay, and we would do the same thing for our other vial, which is dry peas and beads. So this has had a change of 0.03, but again, we have to subtract our 0.02, and so we end up with a corrected difference of 0.01. Our corrected difference numbers are going to be the ones that we graph. So if we look again at 10 minutes, now the reading is at 0.90 from the beginning. So this is another key point, is you have to go back to the beginning because we're looking at cumulative change. From the beginning, there's been 0.03 change. we're going to use that number to correct our other two vials. So in this vial, 0.91 minus 0.77, there's been 0.14 change, but we need to subtract the change through the glass beads. So that gives us only a 0.11 for our corrected difference. Do the same thing here. 0.92 minus 0.87 gives us 0.05. Subtract what was due to just the beads alone, and that gives us a corrected difference of 0.02. You're going to go on and do that for your 15-minute and your 20-minute reading. Remember, you're always looking back. Sorry. Hang on. You're always looking time 0 and figuring out your cumulative change. Okay? The bottom part of the table is the same idea, except this is done in the ice bath. So we just have new sets of data. Here's your 5-minute reading. This is the change in the beads. This is what we're going to use to correct our other two. So this had a change of 0.04. We're going to subtract out the beads amount. It's just 0.03. This had a change of 0.01. We subtract out the beads amount. It's just 0, etc. Fill in the rest of that. Okay, so take a minute to kind of chew on that, practice filling it in. When you think that you've figured it out, then go on to the website, check the key. I posted the key to the Prelab. If your data is correct, then you can go ahead and graph and do the rates. Don't look at everything on the key. Just check to make sure that your table is correct. Then you can go on and do the rest of it, and then check the key for that. And we can talk about this in class also, but hopefully that helps you see what's going on with this data table.